We found some of the same changes in other tissues, however. So the rate-limiting step in glutathione biosynthesis is GCL, and we found that the levels of this biosynthetic enzyme are higher in the Ames dwarf, at least at a couple of ages. We also looked at GGT, which is the enzyme that's responsible for breaking down glutathione into amino acids, single amino acids again, and we found that GGT activity is actually suppressed in the dwarf. So the dwarf is in these tan bars, and it's suppressed across all three age groups that we looked at. And again, 80 to 90% of catabolism occurs in the kidney, so we found this quite significant. And most of the de novo synthesis of GSH occurs in the liver, so we also thought that this was quite important. When we looked at the ability, or to look at the level of activity of the glutathionase transferase family, which is actually a huge family of proteins that have somewhat different functions with regard to substrate utilization, we found that the activity of GST is significantly elevated in these animals, which may contribute to their ability to be more stress resistant because they can detoxify better. So we thought that was significant, and I'll come back to this GST story in a few minutes. So I just told you about glutathione levels, GPX levels, GSSG, GST. These are all, and GCL are all elevated in the Ames dwarf, suggesting that biosynthesis and utilization are increased in this animal, whereas degradation or catabolism of GSH is suppressed in the animals, which may lead, we believe, to higher levels of glutathione available. So we teamed up with Eric Euthys, who's at the Human Nutrition Center on campus, who has a distinct interest in methionine metabolism, and we began to look at the effects, or how the dwarf expression of their metabolic, or methionine metabolic pathway. What we found was this is a series of enzymes involved in the methionine metabolism. So M-adenosyl transferase is up over 200% in the Ames. GNMT is glycine N-methyltransferase, which is a DNA methyltransferase. So it takes a methyl group off S-adenosylmethionine, and that becomes S-adenosyl homocysteine. This was up over 90% in the dwarf liver. CBS is cystathionine beta synthase. So let me back up. Homocysteine can go one of two directions. It can go down an irreversible pathway called transsulfuration, or it can be recycled back to methionine upon exposure to methionine synthase. And so the cystathionine beta synthase and cystathionase that break down homocysteine to cysteine were also increased in these animals, whereas the recycling of homocysteine back to methionine was significantly suppressed. Again, suggesting that the transsulfuration pathway was the primary pathway used by these animals with regard to producing cysteines. And this is just the S-adenosylmethionine to the SAH ratio, which is low, which in the literature, if you look at that, just like the GSH to GSSG ratio, when they say it's low, that's not good. But these animals do really, really well. And I'll tell you, we'll talk a little bit more about that in just a second. So, knowing that this particular system was altered, we decided to look to kind of validate those enzyme levels, those changes, to look at methionine flux. So to look at the flux of methionine through that pathway, you inject a couple different radioactive S-adenosyl methionine. One's tritiated, one's 35S labeled, and then you measure the amount of radioactivity after a certain amount of time that's left in those tissues. And we found that in the Ames dwarf liver, both the tritiated form and the 35S labeled SAM levels were much lower, by I think 56 and 64% lower, which suggests that transmethylation and transsulfuration are significantly increased in the Ames dwarf in comparison to the wild type. And we did find that the 35S labeled SAM was also lower in the brain and the kidney. We didn't look at muscle. So it wasn't just the liver that was being affected, it's other systems that are also expressing an atypical methionine metabolism in these animals. And then if you look at the gene level, looked at message expression for all of those different enzymes involved in both the methionine metabolic pathway and transsulfuration. And again, it validated the data that we saw with regard to protein and activity levels of these enzymes, showing that indeed message expression was enhanced in the Ames, with the exception of a MAP2A, which is a different form the adenylyl transferase, which is not found in the liver, so there wasn't any difference between wild type and dwarf. And the MTR, which represents the methionine synthase, so the one enzyme activity that was suppressed in the recycling of homocysteine back to methionine, the gene expression is also significantly lower. So this was very supportive data of what we had found with regard to activity levels. So the logical thing to do is then add back growth hormone to the dwarf animals and see what happens to methionine metabolism. So that's shown on the right. So if you give back growth hormone just short term, collect the tissues and look again at these enzyme activities, we got a suppression in several of the enzymes with regard to methionine metabolism. We also got a suppression in the GPX activity, which we had shown before, and just looked at it again just to be sure. We found that it suppresses GPX about 70%, suppresses GST activity, and it actually causes an increase in the degradation of GSH in the kidney. So the growth hormone is having opposing effects, which kind of made sense because our particular animal that we're looking at is growth hormone deficient. So we went on to look a little bit further at the different GST classes. With message expression right now, we're setting up some of the specific enzyme assays because they take different substrates. Right now, we found that these are the different classes of GST, so it is a huge family of proteins. The primary ones in the mouse and the human are the alpha, the mu, and the pi classes. They all are involved in xenobiotic metabolism, which we've talked about with regard to detoxification. Cell cycle regulation, they're involved in oxidative stress and apoptosis, in addition to eicosanoid synthesis and steroid synthesis. So we wanted to look more specifically at the different forms and found indeed the alpha class, A1 and A2, are significantly elevated in the Ames. And the A3 and A4 classes are all significantly elevated. This is all liver tissue right now that we're looking at, but we're also going to look at kidney and maybe muscle. So the whole alpha class is elevated. The mu class, the M1 and M3, are significantly elevated in the Ames in comparison. And again, these different classes use slightly different substrates in the liver and the kidney. And then the GST theta class was elevated. And then we found kind of an anomaly in comparison to our other work, where we found everything was upregulated. The GST pi, or GST P1 and P2, were actually significantly suppressed in the Ames dwarf in compared to wild type. And I'll come back to why we think that's important here in just a couple of minutes. So the rest of the GST classes that we looked at were significantly elevated across almost all ages in the Ames dwarf and the liver. So I've told you and showed you evidence that the dwarf has an atypical methionine metabolism. And specifically, S-adenosylmethionine, S-adenosylhomocysteine, and the DNA methyltransferase gene are significantly altered. And one thing to mention is that we look at the liver because 85% of all transmethylation reactions occur in the liver. So this is a very significant tissue for DNA methylation in general. So very naively, we thought, oh, methylation must be different in these animals. And so we took a peek with a collaborator at hepatic global methylation, and we're very disappointed not to find any differences between the wild type and the dwarf. When you dive into the literature, so we're very naive out here, and you dive into the literature and start learning more, you discover you're not going to see anything if you look at global methylation, because it's actually a change in methylation patterns over time that occurs, or you get changes in specific promoters, methylation of specific promoters over time. So we my collaborator set up a very labor-intensive, non-reproducible assay called restriction genomic landmark scanning. Had a lot of troubles with it. Discovered that indeed what we decided was that the dwarf was differentially methylated. The DNA was in different areas. Before we continued with this particular assay, DNA methylation arrays became available for the mouse. And so we sent away those. They go off to Nimmelgen Roche. They have a beautiful system. And you prepare the DNA, send it to them, and they send you back the data. We sent 3 dwarf and 3 wild type liver DNA samples to them. And in our primary scan,